Today's topic is primary cell culture. What is meant by primary cell culture? Primary cell culture is a technique in which the cells are cultured. It's a culturing technique where culturing of cells takes place. It's a culturing technique which is carried out after the isolation of cells from tissues, where the cells are isolated from tissues. So after the cells are isolated from the tissues, the culturing technique which is undergone is said to be as a primary cell culture. And the culturing technique till the next or the first subculture of the cells is called primary cell culture. Means whatever the culturing technique which was done after the isolation of cells and before the first subculture of the cells, such type of culture is said to be as primary cell culture. This primary cell culture consists of variety of differentiated cells. It consists of variety of differentiated cells. Example of these differentiated cells are fibroblast, lymphocytes, macrophages, epithelial cells. Fibroblast, lymphocytes, macrophages, epithelial cells are the examples of these differentiated cells which were grown under primary cell culture. Next we will see the characteristics of before going into the characteristics, here you can see the example. This is a lung tissue from which the cells are isolated. So this is a lung tissue which is made up of cells. From this a cell is isolated and that cell is grown in a petri dish in culture media. And after growing this cell will rapidly divide and it produces a number of cells, and when the petri dish is filled with the cells, these cells shows a contact inhibition between these two. So here you can see the contact inhibition. So if the contact inhibition is seen, such condition is required to change the cells to a fresh media. Means when the contact inhibition is seen in the petri dish, the cells to be transferred to the new culture media, and the transfer of cells from this media to a new media is called as secondary cell culture, or we can also say it is a first subculture. Okay, so the process which was undergone before the subculture and after the isolation of cells from the tissues is said to be as primary cell culture. Okay, next we will see the characteristics of primary culture. If we see the characteristics of the primary culture, here for primary culture, embryonic tissue is more preferred. Embryonic tissue is more preferred than adult tissue, because embryonic tissue can be easily disaggregated. Means easily it can be broken down and it has more viable cells. Next characteristic is the quantity of the cells for primary culture should be taken high, because quantity of cells should be taken high because The survival rate is very less during primary culture. Survival rate is very less. For the reason, quantity of the cells for the primary culture should be taken high. And the cells to be selected with a minimum damage. Cells with a minimum damage should be selected for primary culture. Okay. And the selection of media is also important. If we see the selection of media, with the serum and without serum, we consider selection of media is also important. Mostly with the serum, we prefer fetal bovine serum. Here calf serum and horse serum is not preferred. Calf or horse serum is not preferred. Next, for the breakdown of the cells from tissue, some enzymes are used. So before primary culture, these enzymes should be removed from the cells. Enzymes are removed completely from the cells and then the primary culture is to be done. So enzymes should be removed before culturing. Okay. So these are the characteristics we have to keep in mind while doing primary culture. Next we will see about the techniques for primary culture. So if we see the techniques of primary culture, we see there are more commonly we see 3 techniques. So what are those 3 techniques? First one is mechanical desegregation. Second one is enzymatic disaggregation. Third one is primary explant technique. So first we will see mechanical desegregation. Mechanical desegregation is the technique which was done for soft tissues. It's more suitable for soft tissues. For example, spleen, brain, embryonic liver and tumors. These are the examples of the soft tissues. If you want to take the cells from the soft tissues, these mechanical desegregation is used. For this, these soft tissues should be carefully chopped and sliced. Careful chopping and slicing is done. After careful chopping and slicing of tissue, while doing this, some cells will spill out. These spilled out cells are collected. This is collected in 2 ways. One is by pressing the tissue in a series of sieves. Sieves will be having some pore size. By reducing pore size, first sieve will be having bigger pore size. Next sieve will be having little smaller, and next sieve will be having smaller than the before sieve. So by reducing the pore size of the sieve, a series of sieves will be taken and the tissues are pressed on it. Tissues are pressed on it. Okay. So while pressing the tissues in the series of sieves, cells will be coming down. These cells will be collected for the primary culture. Next collection is forcing the tissue fragments through a syringe and needle. While using a syringe and needle, pressing the syringe, we will collect the cells. So this is one method of collecting cells from the soft tissues. If the tissue is plenty, only we can use this method, because there is a risk of damage of cells while doing this. So this method is having a risk of damage of cells. So plenty of tissue should be taken. Okay. And if we compare this method with the second method, that is enzymatic method, viability is also less. There is a chance of death of cells. Viability of cells is less when compared to enzymatic method. Okay. So next we will see enzymatic disaggregation. Enzymatic disaggregation is mostly preferred. Enzymatic disaggregation is mostly preferred when a high recovery of the cells is required from the tissue. So if we want more number of cells from tissue, we use this method. We use enzymes. So the enzymes which are used in this method, most commonly used enzymes are trypsin, collagenase, and some other enzymes. Okay. So these three enzymes are most commonly used for disaggregation of cells from the tissues. So for separating cells from the tissues, these enzymes are used: trypsin, collagenase, and some other enzymes. First, we will see about trypsin. The process of using this trypsin to break cells from the tissue, that process we will call it as trypsinization. Here, this trypsin, crude is preferred than pure, because crude contains some other proteases also which can easily break the cells. Why? Because crude is used. Why? Because the crude is used. Crude is more effective. Cells can tolerate crude than pure. Cells tolerate crude. Trypsin. Next, it can be easily neutralized in serum. Easily neutralized in serum. If the serum free media is used, trypsin inhibitor is used. Trypsin inhibitor is used in serum free media. And this is easily neutralized in serum media. Okay. So for that reason we select crude trypsin than pure trypsin, and this is also less toxic. Crude trypsin is also less toxic, and this is specific in its action. Okay, so now we will see the process of enzymatic disaggregation of trypsin. So while using trypsin, the tissue is selected. Tissue is selected, and this tissue is chopped for 2 to 3 size pieces. After chopping, two types of processes may takes place: warm trypsinization or cold trypsinization. So there is a different processes for warm trypsinization and cold trypsinization. First we will see the warm trypsinization. Warm trypsinization, if we see, in warm trypsinization, after chopping, the tissue is washed with DBSS, direct dissection basal salt solution, and after washing, it is transferred to a flask containing warm trypsin. So flask with a warm trypsin is taken, and this warm trypsin is maintained at the temperature of 37 degrees Celsius. To this warm trypsin flask, we will transfer the chopped tissues after washing with the DBSS, that is dissection basal salt solution. Dissection basal salt solution. Next, contents are stirred in the flask for every 30 minutes. Contents are stirred means the cells are stirred in flask for every 30 minutes for 4 hours. Next, cells are counted. Cells are counted and it is transferred to media for primary culture. So using trypsin, we are breaking the cells from the tissues. So this is a method of warm trypsinization. Next we will see the cold trypsinization. In cold trypsinization, what we will do is, after chopping, again here also we see the washing step. Next, after washing, these cells are soaked in cold trypsin. These cells are soaked in cold trypsin for 6 to 24 hours. Next, trypsin is removed from the cells and discarded. Trypsin removed. Next, tissue pieces are incubated at 37 degrees Celsius for 20 to 30 minutes. Incubation is done. Okay. Next, cells are dispersed. Cells separates, and again this also transferred to the new media for primary culture. So this method is warm trypsinization using warm trypsin, and this method is a cold trypsinization which uses cold trypsin. Okay, and here you can see the cold trypsin is not suitable for the large quantities of the tissues, that we should keep in mind. Next we will see the limitations of trypsinization. If we see the limitations, Trypsin may damage some cells, such as example you can see epithelial cells, or it may be ineffective for certain tissues. Ineffective for certain tissues, such as means it do not show any effect on certain tissues to break, such as fibrous connective tissue. It cannot be broken down by using trypsin. Next, because for these reasons other enzymes are used in place of trypsin. Okay, so these are the limitations of trypsinization or trypsin. Next we will see okay, next we will see the enzyme that is collagenase. Collagenase. Collagenase is a structural protein which is present in the higher animals. This is a structural protein. This is present in the extracellular matrix of connective tissue and muscles. It is mostly present in the connective tissues and muscles we see. Okay, and here also the crude enzyme is preferred. Crude enzyme is preferred than pure enzyme, because this crude enzyme is also contaminated with certain proteases. Okay, next here the steps involved for doing this is first the desired tissue is taken. And this is suspended in basal salt solution and antibiotics. After soaking or suspending in basal salt solution and antibiotics, this is chopped. The tissue is chopped. And after the chopping process we see the washing step. After washing, this tissue pieces are suspended in a medium containing the enzyme collagenase. So again suspending in collagenase. After suspending in collagenase, this is incubated for 1 to 5 days. Next, the tissue pieces are dispersed by pipetting. So after pipetting, the cells are separated and used for primary culture. And these are used for primary culture. Okay, so this is what happens while using collagenase. The tissues preferred mostly for this enzyme the tissues preferred are human brain, lungs, and several epithelial tissues. So these tissues are preferred. The desired tissues are human brain, lungs, and epithelial tissues. Sometimes enzyme hyaluronidase is used for helps the collagenase and makes it more effective. The enzyme hyaluronidase. Okay, so next we will see the primary explant technique. primary explant technique is the one which was developed by Harrison. Harrison in 1907. This was undergone several modifications and it is still in use now. Now we will see the procedure. Here also we select the tissue. The tissue is kept in BSS and it is chopped in BSS. Next washed. Next BSS is removed. After washing, BSS is removed. Tissue pieces are spread on growth surface. Spread on growth surface, and these are incubated for 3 to 5 days. Medium is changed frequently. Frequent medium change is required when the outgrowth of the cells is observed. Outgrowth when observed, these explants are transferred to a new culture vessel. New culture vessel where the primary culture takes place. So this is the method. Here the explant is developed first, then this explant is grown as a primary culture. So such process we'll call it as a primary explant technique. So these all are the methods of primary culture. Here we can the main advantage of this primary explant technique is we don't need a more or large amount of tissue is not required. Small tissue is enough for the primary culture because there is no risk of cell damage here. Here there is no risk of cell damage. Okay, cell damage is very much less when compared to first two methods. So after growing the cells or breaking the cells from the tissues, one important step is to separate the viable cells with the non-viable cells. So the separation process is very important. This is done using viable cells and non-viable cells are separated using centrifugation. Okay, so this is about primary cell culture. I hope you understood. Thank you.